Today we're going to talk about monohybrid crosses. We're going to go through a bunch of different problems. It's going to review the vocabulary that we've already covered, and I'm going to give you the tools that you need in order to solve monohybrid crosses. So let's get started. So there are 5 steps to solving any type of genetics problem. And the first step is to determine the genotypes of the parents or use what is given in the problem. Sometimes the problem gives you the genotypes of the parents, which is great. Other times you're going to have to figure out the genotypes of the parents, or the letters that are going to represent the genotypes based on the problem. So a little bit more about that. You assign letters for the genotype. So you use letters to represent genotype. The first letter of the dominant trait is usually used when you're assigning letters. So a capital letter represents dominant and a lowercase letter represents recessive. So if we're looking at, if you recall when we talked about Mendel's pea plants, tall was dominant to short. So we would use capital T to represent the tall dominant trait and a lowercase t to represent the short recessive trait. And the capital T and the lowercase t, this letter combination right here, if you recall when we talked about vocabulary, represents genotype. Perfect. So once you've determined the genotypes of the parents, you are going to set up your Punnett square. And the Punnett square represents the possible gamete combinations that can be formed and the resultant offspring. So a Punnett square looks like, especially for monohybrid crosses, is a square that looks something like this with 4 boxes in it, and the letters that you put up here, one on this side and then one along the sides, represents the possible gametes that can be formed from mom. So we could say mom's possible gametes will be represented over here, and dad's possible gametes will be represented over here. So once you've set up your Punnett square, you're going to fill in the squares. And the squares represent the possible combinations that could occur during fertilization. Now remember, fertilization is the union of sperm and egg. So we're representing the possible combinations that could result in the possible zygote of a new individual. Once you've filled in the squares, you're then going to find the genotypic ratio of the offspring. So you're going to look at the letter combinations and group any letter combinations together and put that in ratio form. And then you're going to look at the phenotypic ratio. So take a look at the genotype, because genotype determines phenotype, and then give me the phenotypic ratios. So those are the 5 steps in solving a genetics problem: determining the genotypes of the parents, setting up your Punnett square, filling in the Punnett square, and then determining the genotypic and phenotypic ratios of the possible offspring. So let's do some review and an example of a Punnett square problem. So on your worksheet or your note sheet, you're going to, for each genotype, you have to indicate whether or not it is homozygous dominant, homozygous recessive, or heterozygous. So remember that homozygous means that both alleles are exactly identical to each other. If it's dominant, that means there's going to be 2 uppercase letters. If it's recessive, that means there's going to be 2 lowercase letters. So for capital A, this is going to represent a genotype that is homozygous dominant. For capital B, lowercase b, this is going to represent a genotype that is heterozygous. For lowercase c, this is going to represent a genotype that is homozygous recessive. Capital D, lowercase d will be heterozygous. Capital E is going to be homozygous dominant. Capital F, lowercase f is going to be heterozygous. And then for the last remaining 3, I'm going to let you do those on your own. Now skipping down to number 2, it asks, for each of the genotypes below, determine the phenotype. So recall that genotype dictates phenotype. So what your genetic makeup is dictates what your physical appearance is going to be. So here we have purple flowers are dominant to white flowers. Purple flowers, or capital P, is going to represent the dominant allele, and a lowercase p is going to represent the recessive allele. So 2 capital P's, these flowers are going to in fact be purple because we have at least 1 dominant allele. In this next one, we have a heterozygous genotype, and these flowers are also going to be purple because we have at least 1 dominant allele. We have 2 lowercase p's here, so this flower is going to be white because we do not have any dominant alleles. We have 2 recessive alleles. Now for this one, brown eyes are dominant to blue eyes. I'm going to allow you to do that on your own. Let's take a look at a sample Punnett square problem and employ those 5 steps that we talked about before. So for number 3, it says, set up a Punnett square for each of the following monohybrid crosses. Determine the genotypic and phenotypic ratio for each. So in pea plants, round peas are dominant over wrinkled peas. Cross a pea plant heterozygous for round peas in a plant that is homozygous for wrinkled peas. So in this problem, we have to determine the genotypes of the parents. The genotypes of the parents are not given, but what is given is our dominant and our recessive allele, which is wonderful. So it says cross a pea plant that is heterozygous for round peas. So that means I'm going to have 1 capital R and 1 lowercase r, and because I have at least 1 capital letter or 1 dominant allele, that's my heterozygous for round peas. And I'm going to cross that with a pea plant that is homozygous for wrinkled peas. So that means I'm going to have 2 lowercase alleles. Now I need to set up my Punnett square. So I'm going to draw out a Punnett square here with 4 boxes. Since I'm only doing 1 trait, I'm only looking at 1 trait, and that's seed coat. So I'm going to put my possible gametes. So I'm going to put a capital R there and a lowercase r for my heterozygous plant. And I'm going to put a lowercase r here and a lowercase r here for my homozygous wrinkle-seeded plant. And then I'm going to fill in my boxes. So I'm going to bring my capital letter down, my capital letter down. The capital letter always goes before the lowercase letter just because that's convention, and capital letters usually always come before lowercase letters in any sort of words. Then I'm going to bring this lowercase r down, bring this lowercase r down, and then I'm going to bring these lowercase r's across, this lowercase r across, and now I've completed my Punnett square. And within my Punnett square are my possible outcomes for my offspring. So my phenotypic ratio, I've got 1, 2 boxes out of 4 that are heterozygous for round peas. So I'm going to write 2 for round, 2 round, and I have 2 boxes that have the genotype for wrinkled peas. So I have 2 wrinkled. So my phenotypic ratio is 2 to 2. You could also express this in percentages. I sometimes like to express it in percentages just because it's easier for me to remember and actually see in my head. So I would write this as 50% round and 50% wrinkled. So that's my phenotypic ratio because I am expressing what these seeds are going to look like, what these peas are going to look like. And phenotype represents physical appearance, physical appearance. So physical appearance starts with pH, phenotype also starts with pH. It's an easy way to remember that. Now my genotypic ratio. We're still going to have 50-50 because we have the same genotypes in two boxes as compared to other boxes, but we're going to write this out as 2 capital R, lowercase r, and then your ratio would be 2 lowercase r, or you would have 50% capital R, lowercase r, and 50% lowercase r. So here you're writing the letter combinations, and in phenotypic ratio, you're writing the actual appearance of what those genotypes are going. Let's complete one more problem together. So in humans, freckles are dominant over no freckles. So we're going to cross a homozygous dominant male with a heterozygous female. So again, we need to determine the genotypes of the parents. Fortunately for us, the alleles are given. So capital F represents freckles, which are dominant over a lowercase f, which represents no freckles. So I'm crossing a homozygous dominant male. So that means I'm going to have capital F, and I'm going to cross that with a heterozygous female. So that would be capital F, lowercase f. So that's going to be my cross. Now I have to set up my Punnett square. So I'm going to draw out a Punnett square here. So now that I've drawn out my Punnett square, I'm going to complete my Punnett square by filling in the possible gamete combinations. So capital F for the male, and capital F, lowercase f for the female. So now we're going to fill in our Punnett square. And here I go, filling in the Punnett square. My phenotypic ratio is going to be 2 are going to be freckled, and I'm sorry. That's not the case. We're going to have 4 freckled, 2, 0, no freckles. Or I would say 100% freckled. For my genotypic ratio, I'm going to have 2 capital F, 2 heterozygous, capital F, lowercase f, or 50% homozygous dominant to 50% heterozygous. So you have an additional 3 more practice problems that I'd like you to complete. And please refer back to this video as well as the steps that we reviewed previously to solve those practice problems. So just to review, from this video lecture, you should be able to identify genotypes. So is the genotype homozygous, meaning that does it have 2 identical alleles, whether those alleles are dominant or recessive? Is the genotype heterozygous? From there, you should be able to identify the phenotype or the physical appearance of an organism based on an organism's genotype. And then you should, from this video, be able to predict the probable outcome of monohybrid crosses, and then give me the genotypic ratio and phenotypic ratio of those crosses. Thanks so much, and I will see you later.